All right, so hello everyone. Welcome to the first lecture for Projects and Seminars, Mobile Genomics. Today we will discuss an overview about mobile genomics and the genome analysis in general. Then we will go step by step over the projects that we are proposing for this course. At the end, you will pick just a single project. So I hope you get the main idea for each project such that you can roughly decide whether sounds like interesting to you or you want like to pick something else. I will go briefly through the logistics of the course, then introduce myself and the mentors for this course. So what's the role of this course? So as this is a projects and seminar course, so we'd like to cover the basics first. And we will cover the basics of genome analysis as this course is related to genomics. But as we mentioned in the course description, we don't require you understanding or have any knowledge about bioinformatics or genome analysis. We will teach you all these basics, basically. And that will help you to understand the speed accuracy trade-off in using computationally lightweight heuristics versus accurate, computationally expensive algorithms. And that's our goal, is to have something feasible for mobile genomics or mobile devices where you can do the computation efficiently over these devices. So students will experimentally evaluate different heuristics and observe their effect on the end results. So there are some heuristics, some of them are good, some of them are bad, some of them just enough or good enough for the use case we have. Some are not really good. They compromise accuracy just to gain speed up. We will see this evaluation, and based on this, we'll get some insights with which algorithm might be better for our needs. And this evaluation will give the students the chance to carry out a hands-on project and to implement one or more of these heuristic algorithms. But at the end, the entire project or the entire course will be with the focus of single project. And you may implement these algorithms in your smartphone or any kind of mobile devices where you don't have large memories or large compute capabilities, for example. That will help the society by enabling on-site analysis of genomic data, especially in this time during the COVID-19 pandemic, where you are doing the computation at population scale for the entire world. And there are some places where you don't have the abilities to access cluster computing, for example, or cloud computing. So this will help many research groups or many projects to do the computation efficiently. What are the key objectives we intend to have in this course? So that is the multiple components, and all of them, I think, will provide benefits to you. So the first thing is basic knowledge in genome analysis. We don't expect, again, to have any knowledge in this, so we'll provide this basic knowledge for you before you start the project. And that is related to dry lab. What we mean by dry lab is the computer science or computer architecture or electrical engineering student would do, which is not dealing with molecules or blood sample and so on, but rather than just computer algorithm, then develop architecture that can perform that computer algorithm quickly or efficiently. We will help you to get technical skills in genome analysis and computer architecture, as well as enrich your critical thinking and analysis by facing several problems throughout the semester. We hope you will not face it, but definitely as developing something new, you'll face several issues. And we hope that you use Google first to search what's the problem, what are the solutions for such a problem before you ask us. That will help you to enrich your critical thinking or how to handle any problem in life, basically. And we will help you to get familiar with key research direction. At the end, we are not teaching you some old science or something that many others already did what you are trying to do, but rather this is part of our research, what we are doing ourselves at ETH or Safari Research Group. So most of the projects we are proposing are part of our research, but we don't expect you to do research. So the things are already well defined for you. We already have well-defined problem that we will solve it. We know where you will reach after 3 months. what are the things that you will achieve if you really following us and attending all meetings, for example, following up with all tasks we assign to you, and so on. At the end of the semester, you will deliver a technical presentation to our research group, to many researchers, where you will present the details of your design, implementation. It could be just algorithm or survey or hardware architecture that you implement. And that will enrich your experience on how to deliver a talk in conferences, for example, or in a research setup. All right, what is the key goal? Basically, we would like to teach you how to efficiently implement one of the key steps in genome analysis on portable devices. These portable devices could be smartphone or just a typical day computer where you have just limited resources, for example. And we will not leave it open to you to pick the step you like, but rather we will give you a very specific step in genome analysis where we feel that you can contribute to. What are the prerequisites or the requirement of this course? As I mentioned, no prior knowledge is required in bioinformatics or genome analysis. And we expect you to have a good knowledge in C, but not that expertise level, good knowledge, which is good enough to start programming. And if you have the sense of programming, then I think won't be a problem to use any programming language. The more important thing, and I would like to emphasize that before joining this course, we hope all of you have an interest in making things efficient and solving problems, not rather just doing the job as we need, but rather to suggest some improvement. It's okay if you don't do it, but we can offload these to the next student who join next semester, for example. But we really like to have students who can suggest that this way might be better. I figure out this way. I did this on my own, and I find this, and that. All right, so who are we? So this course is given by Professor Onur Mutlu, who's a full prof at ETH since September 2015. He was, before that, a professor at CMU in US, starting from 2009, and he's still adjunct professor there. He got his PhD from UT Austin. He worked at several companies where he started his computer architecture research group in these companies. And for more information about his research group, you can follow these links. If you'd like to contact him, you can reach him directly over his Gmail account or Gmail address. And he's leading Safari Research Group. And what we are doing in Safari Research Group is several research interests, starting from computer architecture, of course, computer systems, hardware security, and bioinformatics, medical application, and so on. And I am Mohammed Alser. I'm senior researcher and lecturer at Safari Research Group, ETH. I joined about 2 years and probably a couple of months ago. I got my PhD from Bilkent University, Turkey in 2018. I worked at UCLA, TU Dresden, and Petronas before I joined ETH. And my PhD was in accelerating genome analysis, so it's very related to what we have in this course. And my PhD advisors were Can Alkan and Onur Mutlu during my PhD time. My PhD thesis received several awards related to the things we proposed in the thesis. So if you'd like to reach me, this is my email, this is my webpage, and this is my Twitter account where I share a few things about genome analysis or research in general. And I'm mainly interested in bioinformatics, computational genomics, or metagenomics for viruses, and computer architecture. I'm especially excited about building new data structures, algorithms, or architectures for several applications, especially making intelligent genome analysis a reality. So if you're still wondering what is genome analysis or what is intelligent genome analysis, I will explain that in a couple of slides more. This course is happening with the help of many great researchers from our research group, starting from Juan, who's senior researcher and lecturer. We have also Damla, who's a PhD student at CMU. She's part of our research group at ETH as well. We have Jeremy, who's a PhD student, and John Fertina, who's also another PhD student. All of these will be mentoring you as a main mentor or as a co-lead for any of the projects that we will assign. You can see from their research interests down below that they have really diverse research interests, and that will enrich the projects that we have where we can get help from several expertise. I would really like to recommend that everyone go and check our website, our Safari Research Group webpage, where you can get more information about each of our members, where you can learn about the research we do or what kind of project we are doing. All right, so what we expect at the end of the semester or throughout the semester. First, attendance is really required because this is very important to maintain a good progress throughout the semester. And this is true for all meetings. So for this meeting, we specify the time, but for the next meeting, it will be between you and your mentors. Probably for each student will be different meeting. And we will meet once a week. The second thing, it's really important to study the learning materials that we already assigned last week. I think most of you or all of you went already through it, at least watching the videos that belongs to each of the research paper we assign. So each student will carry out a hands-on project, as I mentioned. It could be building or implementing or even coding and building designs for the project. Depends on the nature of the project. The participation, we will have a GLIB group, which is similar to WhatsApp, but we use it for research purposes. For each project, we will have a GLIB group where we discuss the things before we meet. So we will meet once a week, but between the two meetings, you may face a problem or you find something interesting or you'd like our help to reschedule the meeting and so on. We will use this for communication for each group. But we still have a Moodle and the email channel if you'd like to reach all of us, for example, at once. At the end of the semester, you will deliver a presentation and a GitHub repo where you share all coding and all stuff in a private mode, because we don't expect that this will be ready for public at the end of the semester. We will help those who did really good progress in their projects to get their work published in good venues. And that will provide a lot of benefits, especially if you are thinking in continuing your studies for masters or other research purposes. This is the course web page. The slides are already online there. We can share the link maybe offline if you didn't have a chance to access it yet. We will share over Moodle and email, so please check it frequently for any announcement, especially when we assign the projects. And next meeting, we will give you a chance, probably starting from today, for a couple of days to select few projects and where you indicate your preferences. And then we will assign a single project to you. And then we will have one-to-one meeting with each student to double check whether you really understand what we expect from the project you select and whether it really matches your skills or expertise in successfully completing that project. So it's very important to study the learning materials before our next meeting, which will be next week. And we will start the projects next week as well. All right, so that's all for all logistics about the course. If you have any questions, maybe I can take it within 5 minutes or so. All right, great. Seems no questions. So now I will just have a brief overview for the genome analysis. What is genome analysis? So as you can see from this picture, that there are many individuals that are really different. They might look the same from outside, but their DNA should be at least different. And in genome analysis, we would like to identify or measure or compare genetic materials such that we can find these differences that make individuals really different. However, we don't have any machine, unfortunately, that can take a blood sample and give you the full context or the full strength of your DNA. So without having this text, we cannot build any computer algorithm that can operate over this text and try to find the differences between two strings, two DNA strings. What we have is this machine, what we call it a sequencer or sequencing machine. It gives you chunks or random chunks of your DNA. These chunks could be short, like 100 character long, or it could be long, like 2 million character long, but they don't cover your entire human genome, which is 3.2 billion characters. And unfortunately, these pieces, which we call them reads, so each read has no information at all about the order or the location, where each piece is located in your reference genome or in your DNA. So it's really challenging to start processing these pieces and try to infer where are the differences. You need to really build your reference genome first, and then you compare it with another reference genome such that we can infer the differences. And to build this, we need to have the data first. What kind of data? First, we have a file, we call it FASTA file, and that's what we call reference genome. It's 2 lines only. In this file is really 2 lines. The first line is a header, which starts with this sign, greater than, and this is just ID for the sample, because you may have many samples, or you do sequencing for a family where each member has its own FASTA file, and that is the ID where uniquely identify this sample. The second line is the genetic material that you have, which is the full complete reference genome. So this is really the 3.2 billion characters of your DNA. And this is, you can see it's just covering ACGTs. You cannot see anything else over, than ACGT. The second file, which we call it FASTQ, which is the things we get from the sequencing machine. And for each segment or each read, we have 4 lines. The first line is the identifier, the header, which is similar to the FASTA file. The second line is the read itself, which is the genetic content that you are looking for. You can see, similar to the FASTA file, we have ACGT contents, but we have a couple of ends here that represent ambiguous bases. It means the sequencing machine could not decide whether this is A or C or G or T, such that it just have it as N. And the third line is just a sign. You can ignore it, has no useful thing at all. The last line is the base quality or the score. How confident the sequencing machine was when it reads this base or any of these bases, for example, the T here, the confidence was F. What does mean F? This is the ASCII code. If you convert into ASCII, you will get in percentage what was the confidence level to read, or the probability of sequencing this correctly or wrongly. So what we have to do is to get the FASTQ file and the FASTA file. The FASTA file will be our reference, same as you solve a puzzle, and you try to stitch these pieces together such that you complete the puzzle and you get the full DNA of yourself. So this is the FASTQ file, and this is the FASTA file. And each read here is a small piece of the puzzle. It could be large, it could be small. Of course, if it is large, it will help us solve the puzzle quickly. Okay, so what's the problem? Yeah, I forgot to tell you that this process, we call it read mapping, because we have these pieces, which each piece is called read, and then we try to map each read to this picture such that we have read mapper. The sequencing machine, which tried to read your DNA, can process 48 FASTQ file in 2 days. However, if you'd like to process this in a computer, you could only process single human genome or single FASTQ file within 32 CPU hours. And you can see, if you would like to do it for the entire Switzerland population, how long you need to do that. Imagine if you need to do this twice a day to check for COVID-19, or 5 times a month, and so on. Okay, so that's about genome analysis. Now, what is intelligent genome analysis? For intelligence part of the genome analysis, we really would like to have a fast genome analysis. So I don't want to wait 10 hours to get my COVID-19 test results, for example, or to get my DNA results. I would really like to have it maybe in 1 minute, a few seconds. And I don't want to waste energy while doing so, because I expect to do it at larger scale to the entire population. So if you require, or if the architecture or the hardware you are using to do the genome analysis is not efficient, then you are wasting a lot of resources. And I have example about this in the next slide. The DNA itself, or genetic data, is very valuable asset. What does that mean? If I share your DNA data with someone else, then I can reveal too much information about yourself, more than you ever expect. So I can know about your family members, the disease they have, what's their hair color, eye color, for example, the disease you have, or the disease you might have, or the disease that your kids might have in the future. So there are too much information can be revealed from your DNA. That's why I don't want to, for example, offload the DNA to the cloud computing and do the processing there, because you are giving too much information. It's not as scary as this, because revealing this information is not easy, but for the experienced people, they can reveal these things. And there are a lot of research about protecting the privacy of your DNA, for example. We want to have this genome analysis process scalable, such that we can do it. If I have single CPU core, I can do the process easily. If I have 40 CPU cores, then I expect to have 40x reduction in the execution time. And that's what we call scalability. So I would like really to have scale over the resources. And I don't want to mess with the accuracy of the results. If I tell you that you have this disease, you should have it. Not the opposite. So this is very risky to compromise the accuracy just to gain some speed up by using heuristics. Okay, so this is today's computer architecture, where we have the sequencing machine here, we have the storage, and then the main memory, then we have several level of caches inside the CPU, and then the processor itself. And if I would like to move the data from here to the storage, all the way to main memory, then to the CPU, then that will consume the single memory request from the main memory, for example, here, will consume at least 160x more energy than a single operation, complex add operation, for example, at the CPU side. So it's really expensive to do the computation plus moving the data between these different components. And for that, we would like to discover some ways where we can do the processing over FPGA, GPUs, or inside the sequencing machine itself. And that lead to an open question, how and where to enable the fast, accurate, cheap, and also privacy preserving and exabyte scale analysis of genomic data. All of these very good research topics for PhD or postdoc or masters, but of course we are not expecting such projects from your side, but you are definitely contributing toward this goal. All right, so the last thing I would like to mention is that most speed up comes from parallelism enabled by both novel architectures and algorithms. So developing algorithms alone is not enough. Developing architectures is not enough. We would like really hardware-aware algorithm where you really know what's your hardware is capable of doing so, and you really like the architecture itself to know what's the algorithm is doing. So by developing both of them, you will achieve the highest or the largest level of parallelism. We already assigned this as part of learning materials, so I would really suggest to go watch the entire lecture if you didn't do so. That will give you a lot of fundamental and basics about genome analysis, which will be helpful to carry out the project directly next week rather than spending some time in reading. These are some of the research papers that we did recently as part of Safari Research Group. So you can go over them. If you find something interesting, let us know. We could change the project, or we could do something else if you're really interested in something else. That's all. I would like to remind you that at the end of the semester, we will offer you to continue working with us at any level of research. It could be just a semester project, could be thesis project, could be master's project, or it could be just a PhD student later on and so on. So if you are at ETH, please send us email we have a different criteria or application cycle. But if you are outside the ETH, please apply through this link, and we will contact you as soon as possible. That's all for this. Next, we will discuss the projects that will be internally. We will not record it. And yeah, let me stop sharing this.